We're just going to give it another minute before we get going, everyone. Let's get started here. On behalf of the Children's Research Board, welcome to our first of 3 Research in Focus calls. My name is Kathy Graham, and I serve on the Children's Research Board and head up the Board Education Committee. Today's topic is very timely and focused on the remarkable COVID-19 research being done at Stanley Manne Children's Research Center at Lurie Children's. Our panel of researchers will be presenting their COVID-19 research funded by Springboard Research Awards that you will learn more about during the call. Our second Research in Focus call will be on October 21st at noon and will explore pediatric asthma research with Dr. Rushi Gupta. The third and final call will be on November 19th and will examine adverse childhood experiences featuring Dr. Nia Hurd-Garris. I want to thank all of our 2020 education champions that will be listed on the next page. Their support has been very, very helpful to the Children's Research Board and of course to Lurie's. I want to let you know that we received a number of questions for the panel from the registration process. These questions will be addressed during the call. And if other questions come up during the call, please use the chat function and the panel will do their best to answer your questions. So it's my great pleasure to introduce the moderator of our panel today, and that is Dr. Patrick, he goes by Pat Seed. He joined Lurie Children's Hospital and Northwestern University in the fall of 2016. He is the Children's Research Fund Professor of Pediatrics at the Ann and Robert H. Lurie Children's Hospital and at Northwestern University. He serves as the Associate Chief Research Officer for Basic Sciences at the Stanley Manne Children's Research Institute and as the Division Head for Pediatric Infectious Diseases. Pat completed his MD and a PhD in microbiology and immunology at the University of Rochester. He also completed residency and chief residency at the University of Michigan in pediatrics before completing a pediatric infectious disease fellowship at Washington University. Before joining us, he spent a decade at Duke University as assistant and associate professor of pediatrics, molecular genetics, microbiology, and urology. A very accomplished individual indeed. Dr. Seed's research program investigates how microbes establish communities throughout the body and how they emerge to produce infections of the urinary tract, bloodstream, and central nervous system, as well as non-infectious diseases such as childhood asthma, growth failure, and neurobehavioral disorders. His lab combines next-generation genomics with human cohort studies, animal models, molecular biology, immunology, and biochemistry to understand their molecular basis and to identify translational opportunities for new therapies and diagnostics. He holds multiple patents for novel anti-infectives, and his work has been generously funded by the National Institute for Health, the Department of Defense, the March of Dimes, and the Gerber Foundation, among others. Pat is the mastermind behind the Springboard Awards that you'll soon learn a lot about. Not only did he create the awards and champion their funding, but he meets with all the awardees on a monthly basis to guide and mentor them with their COVID-19 research. So, Dr. Pat, please take it away.
Great, thanks so much, Kathy, for the generous introduction. So thank you everyone for joining us today. This is really a pleasure for you to tell you about some of the work that we're doing related to COVID-19. Before I get there though, I wanted to set the stage for you and really tell you about the remarkable forward momentum that we have at the Manne Research Institute right now. It's been an incredibly productive year, and despite all of the challenges of the pandemic and not much before that, a recent move into our new research institute, we really had incredible momentum towards our overall mission to improve pediatric health through advanced scientific discovery and translation. So if I can get the next slide, that'd be great. Thanks. So I think you'll see that we had quite a bonanza year. In this past year, we published over 1,100 manuscripts. And just between March and July of this past year, really when the pandemic was in full swing, our investigators applied for over $131 million in grant dollars, and we received over $19 million in new and continuing grants, over 165 of them during March and July as well. And many of our investigators moved ideas and technologies through the stages of patenting from provisional to full patents during this time. It's really been an impressive year. If I can get the next slide. We've also had a really large exposure to the community to disseminate and share the work that we've been doing and help to best inform our communities through this outreach that we've done. Our news releases have been picked up in a wide variety of very high profile journals and media sites, and have resulted in a tremendous amount of value and dissemination of these ideas out to the community. The next slide. And what's really remarkable is that this really is a very broad-based effort throughout Lurie Children's and the Manne Research Institute. We've had investigators from all of our departments that you can see over on the left-hand side. And as examples, even within the Division of Department of Pediatrics, we've had 19 divisions and 7 divisions in surgery who are all represented in the type of work we've done. And our work ranges from population health, where we ask how social determinants of health and disparity and policies all intermix to have different health effects and outcomes, to clinical trials that range from the very first studies in kids, including remdesivir as an example, where we were one of the first centers to administer that to children for COVID-19, all the way out to studies of drugs that have already been approved by the FDA, and we're looking in the communities to see how do those drugs work for children to help improve health. We do a lot of basic science. You're going to hear about some hints of that today in preclinical science. And we do a large amount of translational science that takes questions from the community or our children in our trials over to the bench in basic science, or takes questions from basic science and moves the solutions and discoveries we find into children for clinical trials to apply them there. Next slide. So today, we have the great pleasure to hear from some of our very talented investigators who have been awarded these COVID-19 Springboard Awards. These awards were intended to be our rapid response of research to the pandemic. They were intended to fund experts who had shovel-ready activities and very potentially high impact types of research that span from our preclinical science all the way through clinical trials and population health. And the investment was about $25,000 per individual. So a good amount of money to get going, but not a tremendous amount. So it shows you how this amount of money can really spark some very important research for us. And we stress multidisciplinary work across these different teams to get many perspectives to what is a challenging problem. This pandemic, we've all seen the many facets that it affects in terms of our community, our families, and our patients' livelihoods and their health. And you're going to see that many of our investigators are asking critical questions about how does the pandemic affect our communities in access to key things that they need, like food, housing, utilities, healthcare, personal economies? What is their response to potentially the availability of a vaccine to COVID-19 when it becomes available? How does age affect the immune response to COVID-19? Why are kids relatively protected and less ill than adults on average? We can learn a lot from asking that question. Do kids make different responses of antibodies to the virus naturally? And can we use information about the antibodies they make to make better vaccines or to make antibodies in the laboratory that could protect other people from the virus? And how does the child's immune system so well handle COVID-19 compared to adults? So you're going to hear about this really terrific work right now. I'm going to hand this over to Jennifer Kuzma, who's going to be our first speaker. And I'm going to ask all of the speakers to give a brief introduction about what divisions and areas of medicine they're in, and then they're going to take you through a brief introduction to their work. You're going to hear that their work's at different stages. Remember, we haven't had long to start to understand these problems, but it's really, they've been making tremendous progress forward. So Jenny, take it away.
Thank you so much. Hi, everyone. My name is Jenny Kuzma, and I am an attending in the Division of Advanced General Pediatrics, so I practice as a general pediatrician. I recently joined the faculty here at Lurie officially in July, before which I was a fellow. And I'm working with Nina Alfieri, who's also a general pediatrician and faculty member on our Springboard Award project. Nina and I set out to ask the question, how has the COVID-19 pandemic affected the health and fundamental needs of children and families in the Chicagoland area? Next slide, thank you. So why were we interested in this? Nina and I both were having the experience of being in clinic and seeing families and hearing a lot of stories about the social impact that COVID-19 was having. We were hearing stories about things like our therapies have been delayed from physical and speech therapy. We can't get into our subspecialists or our generalists. People were concerned about their child's development. All these children were suddenly at home and no longer having those social interactions. And then economically, a lot of families were very hard hit with furloughs, food insecurity, housing insecurity, issues with paying their utilities, and what to do with childcare when daycares were closed. And then there was also the mental health impacts that we were concerned about with anxiety around what was COVID-19 and how is it going to impact their family. So we really wanted to take a look at both the health needs and fundamental needs that families in Chicago were experiencing. So to do this, we implemented a survey of Cook County families through Qualtrics, which is a web-based, sophisticated survey platform where families could complete the survey on their phone, computer, tablet, whatever they had that could access either data or Wi-Fi. And we were able to send out the survey to Cook County parents, anyone who had a child 18 years of age or younger. And we asked these questions on all those domains I was talking about on the previous slide. And then also we're able to ask questions about, did you agree with stay-at-home orders? And if there was a COVID-19 vaccine, would you get it for yourself or for your child? And we really, what was nice with Qualtrics survey mechanism is we were able to send out the survey broadly to all of Cook County and also make sure that our respondents, the demographics of our respondents were consistent with what we thought the makeup of Cook County's population was based on the most recent census data. So in the end, we had 1,425 high quality responses. And I put a little bit of the makeup in these pie graphs for the slide just to show you that we truly had a racially and ethnically diverse population as well as socioeconomic level and various insurance types represented as well. So why do we do this and what is the outcome that we're looking at from this survey? So really our goal was to better understand the needs of the families that we serve in clinic and throughout Cook County as a representation of Chicagoland. And got really rich information about where these families found that their needs were deficient, and what they thought where their gaps were as they've experienced it. So instead of us guessing where there was gaps in their services or in their needs, we really got answers that are concrete in what these families need. It also is going to allow us to better understand and predict COVID-19 vaccine acceptance and hesitancy. So what populations or what pockets of Cook County are more hesitant about the vaccine, and where can we really target educational materials when the vaccine becomes available, to make sure that we are really reaching out into the community. And the other great thing about this Springboard Award and getting to do this project is we partnered with Lurie Children's Hospital Healthy Communities. So once we have some of these results from the survey and have finished analyzing our work, we'll be able to actually reach out into the community and apply interventions based on what we find that these families need. And I will turn it over to Dr. Coates.
Hello, helps if I unmute. So I'm Bria Coates. I am an attending physician in the Division of Critical Care. And I am a little bit more on the basic science side of things. So we'll talk a little bit today about our research into the mechanisms of disease and COVID-19. You can go to the next slide. So my background, my research interest has been in why children get so sick from viral respiratory infections. So in general, we think of things like influenza and another common virus, RSV, as disproportionately affecting children, young children, and elderly adults. And my research has focused on what is it about the initial immune responses to this virus, these viruses, that might impact how children manifest their symptoms. And our research in RSV, or in influenza using a mouse model, has led us to the conclusion that it's not an inability to control the viral infection, but instead we see differences in the immune response to the virus. So this is just a depiction of our data in mice with influenza, where we show over here for survival, no matter what amount of virus we give the animals, the young mice die at a higher rate than the adult mice, so similar to humans. But this is showing viral load, that they're able to get rid of the virus at the same rate as the adults. So they're dying despite getting rid of the virus. And we've shown that this is due to, in part, some of the initial responses to the virus by the immune system that brings cells into the lungs that perpetuate damage even after the virus is gone, and that if we block some of those processes, we can actually save the juvenile mice from influenza. So interestingly, with COVID-19, what we're seeing is the opposite, that children are not getting as sick as adults. And so we felt like we were in a position with this new virus to investigate that problem. We could go to the next slide. So this is how we're attempting to do that. So in mice, we can just take out their whole lungs and look at the immune response and look at the virus. In children, obviously, that's more challenging. And so what we had been developing is a way of assessing the respiratory response to viruses in kids. And we have been using a small pliable plastic probe to scrape the inside of the nose, and with that we're able to get the cells that are infected by the virus, along with some immune cells that are recruited into the area. We put this directly into a little vial that breaks apart the cells in the virus and preserves the RNA. And we are able to then purify both the host RNA and the viral RNA. And when we sequence that, that gives us an idea of how the cells are responding, how much virus is there, and how the cells are responding to the virus. And we can combine that information with what we know about the children from their charts, which then gives us a pattern of gene expression in each patient. And that will allow us to compare potentially how the younger children respond to the virus compared to the older children, and how children with different other medical problems respond to the virus compared to others, and how children with a lot of virus in their nose compare to children with less virus in their nose. And so far we have about 96 samples that include children and their parents that we enrolled as they came through the hospital this past spring and into summer. We just finished the sequencing. So I don't have any data to show you from that, but just to give you an idea of what it might look like, on the next slide is what we did with a pilot study in RSV. So these are children who were admitted to the ICU with RSV, which is a virus that is the most common admission for children under 2 to the ICU. And what we found was using this process was that on the day that children came into the ICU, if we scraped their nose, the children who would go on to have a quick recovery and leave the ICU within a couple of days, their gene response in the nose looked very different just on that first day of ICU admission than the kids who would be in the ICU for up to a week. And these are just an example of the sorts of genes that we saw expressed in the children who would take a long time to recover from their RSV infection, and the types of immune responses that were elevated or decreased in the children who had a slow recovery from their RSV infection. So this is what we're hoping to be able to define these patterns in the children that we enrolled with SARS-CoV-2 or the virus that causes COVID-19, so that we can pinpoint how the children are responding differently depending on age, other medical problems, etc. All right, you're up, Anne.
Okay, well, good afternoon, everybody. I'm very grateful to be able to discuss our research with you a little bit today. I am an attending physician in the Division of Infectious Diseases. I've been at Lurie Children's for a few decades, actually. I think that all of us who've received these Springboard Awards really are very grateful to be able to make some contribution to trying to solve this terrible pandemic that we are experiencing. So it's really an honor to be able to do so. Next slide, please. So our work, if you think about it on a very basic sort of level, we know that everybody pretty much makes antibodies following an infection, no matter what kind of infection it is. And we make antibodies because they help us recover from the infection. They often will protect us from getting that infection again in the future. And sometimes when you identify what antibodies people make, it can be helpful occasionally for treatment and often for diagnosis. And as we've already heard, and you already, I'm sure, recognized, children are often asymptomatic even when they have infection with SARS-CoV-2, the virus that causes COVID-19. So there could be many different sorts of explanations for that, but what we are investigating is whether there is something about their antibody response that is to the infection that's different than in adults who seem to get more serious disease. And if we did identify that that was the case, the idea would be that maybe the antibodies that children make could be more useful for treatment than perhaps those from adults. It could provide clues about how we should make a vaccine that would stimulate that kind of an antibody response. And it might also help us to make even better diagnostic tests than what we have. Next slide. So our project is to examine the antibody producing cells in the blood. And this is kind of a sophisticated sort of technique. There have been a lot of advances in what's called molecular immunology, so the immune system using molecular biology methods, and we're able to use those new technologies to identify single antibody producing cells in the blood that have arisen in response to infection with this virus. So we look for those plasmablasts that are so-called plasmablasts, which are the antibody producing cells. We look for the ones that are particularly prevalent in the blood of children who are about a week after having their acute infection. And what we do is, once we've identified those, we make them in our laboratory. We examine them further to find out exactly what part of the virus they're targeting, that they're trying to combat. And then we can go back to just regular methods where you just take some blood from a child. You don't have to do any fancy plasmablast work. You can just test for those antibodies. And we could also do that from adults. Next slide. So what we want to learn from these studies, we have some preliminary data, and we're going to get some additional data before we draw any conclusions. But what we hope to learn is what different kinds of antibodies children make following this infection. We will have generated a group of antibodies that we think are particularly important in children's response to the virus. We'll know what they're exactly, what part of the virus they're exactly combating, and see how over time in the blood of children who've been infected, what kind of a response we have to those proteins, the pieces of the virus. And of course, the other purpose for this is to get some preliminary data that will allow us to do further studies by submitting other applications for additional grant funding to outside agencies, and learn more about the differences in the antibody response between children and adults with this virus infection. We also would like to extend the study to the studies of the children with the inflammatory syndrome following COVID-19 that I'm sure you've all heard a lot about. So that's a future goal. And I believe that's my last slide, and I will turn it over to Taylor.
And so, one of my lights just went off, apologies. So my name is Taylor Heald Sargent. I am a new assistant professor. Similar to Jennifer, I just finished my fellowship at Lurie and was able to stay on as an assistant professor in the Division of Infectious Diseases. I'm actually doing my first attending week next week, so I'm very excited to get back to clinical service. In the meantime, though, I've been trying to focus on research, specifically a lot looking at COVID-19. And as both Anne and Bria mentioned, something that we find fascinating is the immune response to COVID-19 in children. Next slide, please. So the interesting thing about COVID-19, and actually this slide in particular, is it's a slide you could have made even before the pandemic started. And it was made, in fact, with SARS-1 infection and the infections in children that occurred there. We actually knew a lot of this data, which was that in the course of clinical infection, you first have what some people have termed as a viral phase, where you have a lot of viral replication, high viral loads, and over time that goes away. But then at the same time, you have control by the immune system, but sometimes overexuberant immune system, and what people have termed the cytokine storm. And as time goes on, that can lead to actual danger and pathology in some individuals, specifically in adults. And it was known from the first SARS virus that the kids were somewhat different. They weren't just little adults, as we like to say in pediatrics, that something else was going on. And it seemed to be that the immune system is where the difference could lie. So going into the pandemic this year, we already knew back in February that we needed to study children. We needed to figure out what was going on and what was different. And we felt uniquely poised at Lurie to be able to answer that question, given how far our reach is in the Chicagoland area and also our laboratory capabilities. Our lab was one of the first in the area to actually get testing going for children and in adults, and to get a drive-through center going. So we knew we needed to capitalize and contribute to the breadth of knowledge about pediatric COVID-19. Next slide. So what a team of us got together to do was set up a biorepository of samples prospectively collected starting in March. So we started to talk about how can we collect samples in late February, early March, and then all of a sudden we were all locked down and it went into overdrive. We had to figure out not just how we could start a prospective study, but how we could do it all remotely. And through a lot of teamwork and really across the board, from regulatory IRB approvals to people on the ground in the hospital helping out nurses on the floor during this stressful time, we were able to collect over and enroll over 30 patients who were admitted to Lurie. We didn't know if we would get 2 or 200, and luckily we're kind of in the lower side. Not too many kids did get sick, but the ones that did end up admitted to the hospital, we were able to collect a wide variety of samples from them across multiple time points. So we were able to collect blood samples, samples from their respiratory tract in one individual who was intubated, their stool, so we can hopefully look at their microbiome, and also some of their urine. And these individuals had samples ranging from 1 time point up to 5 different time points that we were able to collect. So we tried to collect samples at prearranged time points throughout their stay at Lurie up to 2 weeks into their admission. Next slide, please. So thankfully, the Springboard Awards came on at the same time that we were collecting these data, and we realized that we were going to have enough samples that we could actually make some useful analyses from them. So this award enabled us to start looking at some of those serum samples that we've collected. We've identified patients that we have at least 1 serum sample from, and this is the range of those patients. So it's a fairly representative age group, from a tiny baby up to a 17-year-old. We didn't enroll any of the older adults or older young adults in our hospital. Slightly skewed towards male Hispanics, and I think that has to do with who was in our hospital. Some of them were previously healthy, but some had a variety of issues. One was just some more mild chronic conditions, such as hypertension or asthma, and then all the way up to having some patients who had cancer or had congenital heart disease that were more severely affected. And some were admitted to the ICU, but some didn't even have symptoms. And now, using the funds provided through this award, we're going to be able to analyze certain aspects of their immune response, specifically looking at some of the floating immune modulators, so chemokines, cytokines, interferons, to see if there's a difference between these children and other collected samples, such as those that were collected from other viral infections at Lurie in previous winters that were prospectively put in this biorepository, and also samples that were collected over at Northwestern from the adult patients who had more severe COVID. And hopefully we can pinpoint what makes it different, what's different in the immune system between the adults and the pediatrics. There are hundreds of clinical trials going on looking at many different modulators or therapeutics targeted at the immune system. It would be wonderful if we could look at the kids and figure out which one of those drugs we should actually be focusing on to make the most difference. That's it, thanks.
Well, I want to thank all of our speakers so much for presenting the work that is well underway, and we're excited to see the next phase of all of this work. We do have a number of questions for you all, and so I'm hoping to take some time now to take some of these questions that all of you were generous to submit to us, and see what our responses are. So let me start with some general questions about COVID-19, and then ask some about the specific work that all of you are doing. So I'll start with Dr. Heald Sargent. Can you talk a little bit about what we know about child-to-child transmission or adult-to-child transmission? How often is it happening in those types of interactions?
I wish I had an exact answer, which is pretty much what the answer is to every question about COVID right now, is we need more information. I think early on, we didn't know, there's questions of, can children transmit to anyone? Can they even get infected? And I think now we definitely know that kids can get infected, and they have a lot of the prerequisites for being able to transmit. They're able to replicate the virus in their nose, and they're able to have it without, replicate the virus even without having necessarily symptoms. And they are in close contact, as we all know, with other people and their behavioral habits. Hand washing, mask wearing, keeping snot to themselves is in general not that great. So all signs would point to their ability to transmit. Now, more recently, in the last couple of months, there have been some studies that have come out that have found that children were actually linked to in transmission chains. So there was recently an article that came out about childcare centers in Utah, where they were able to pinpoint children that contracted the virus there and then brought it home to adults. But the big unanswered question is, how often is that happening? Are children getting infected as often as adults? And are they able to spread it as well? We really need larger scale studies to be able to figure it out. And until those can be done, I think we need to continue being as safe as possible.
Yeah, great, thank you. Dr. Rowley, do we know anything about kids who get COVID-19, or even adults for that matter, what the long-term effects might be?
There's another good question that we don't have all the answers to. I think it's very important to follow the patients long-term. You know, recently there's been a lot of publicity about potentially long-term heart complications in patients who recover. We don't know if that's really true or not, and we need to follow up on that. Certainly most people who recover from COVID-19, they complain often about being very tired for a long time, but otherwise they seem to do pretty well. But I had listened to a grand rounds that Tony Fauci gave a couple weeks ago at Harvard, and you know, one thing he said is that, you know, we all need to stay humble because we think we know the answers to a lot of these questions, but sufficient time and research has not yet been done to be able to give the final answer to all these questions. So it's a really good one, but we need more time and more data to really have a precise answer.
Great, thank you. Dr. Kuzma, there's a few questions about particularly babies. Do you, in your practice, do you advise that babies have masks that are put on? Is that something that they need to do?
Yeah, so that's another really great question. The recommendation that has come out from the American Academy of Pediatrics, and also what we practice here, is that children under the age of 2, we do not recommend wear masks. It's not because we don't want them just as protected, but it does pose a significant choking risk, and they're just not really all that great at wearing them and not actually just like chewing on them and putting them on their mouth, in their mouths, or actually using a mask for the purpose. But it is, kids over the age of 2 do a really good job of wearing their masks, and so we recommend ages 2 and up wear them, but for babies it's pretty hard to do.
Great, thank you. Dr. Coates, there's a question about do young adults who have congenital heart disease, do they have an increased risk of more severe COVID-related disease? And I guess I would amend that and say, as an intensivist, somebody who works in the intensive care, who are the groups that you think of that have the highest risk among young adults, adolescents, or kids?
Another really great question that doesn't have a definitive answer yet, though it seems like there are certain groups that we generally worry about with viral respiratory infections, including congenital heart disease, and then you add the age piece, so that the teenager with the congenital heart disease may be at higher risk. But in our institution, we haven't really seen that bear out. The other children that we worry about are immunocompromised children or children on immunosuppressant medications. And again, we've been lucky in that that also hasn't been a big problem at our institution, but I do know there are, some other children's hospitals who have had some children in those categories who have had a trouble getting rid of the virus once infected. The other categories of children that we worry about are children with chronic lung disease or with neuromuscular disease who have also tend to carry higher risk for viral infections. But the data hasn't quite borne out yet. So still, I think a lot of things are going against our general patterns. And we haven't seen those groups having more severe disease yet.
Great, terrific. Dr. Rowley, I hope everybody's impressed or understands how amazing it is that you can make artificial antibodies in the laboratory. Can you talk a little bit about, are there data that people who get antibodies to COVID-19 when they get infected, are they protected, or is there any reason to think that the antibodies people are making may not protect them against future infection with the SARS-CoV-2 virus?
Yes, so that's a question we'd all like the answer to. I think what's been observed so far is that, shortly after having the infection, antibodies are pretty high in the blood in most people. And then they tend to fall over time. Now that happens actually in almost all infectious diseases. We have our highest antibody titers shortly after the infection. And then when we don't need, our immune system hasn't been stimulated so much because we don't have the infection anymore, the antibody levels tend to go down a little bit. And so the question really is, and so it's been observed that they go down, and some reports have been that they go down a whole lot, which has made investigators concerned about whether that means you might be susceptible again. But we have plenty of kind of other infectious diseases where that's happened and the antibody levels go down quite low, and yet it still seems that the person doesn't get the infection again. And that might be because of other arms of the immune system helping out, or it may be that what we think is a protective amount of antibody really isn't, you don't need it that high. But all of these are unknown. There's been some concern because it had been noted with the usual kind of mild coronavirus respiratory infections that antibodies don't persist very well, and you can get repeated upper respiratory infections with coronavirus through your whole life. So there's concern about that. But it's interesting to note that there's a pretty close relationship between SARS-CoV-1 that not that many people got infected with, and SARS-CoV-2, and it sort of has appeared that people who had SARS-CoV-1 so far don't seem to get SARS-CoV-2 very often, but the numbers are low, so we still can't say that's absolutely true. So again, another good question we need more information about, and we don't know for sure what level of antibody is necessary to protect you from a future infection.
Great, terrific. Dr. Kuzma, you're going to get this, very rich set of data from a lot of different parts of our community in the Chicagoland area. What do you do with the data, particularly regarding attitudes around the vaccine? If you see areas where there's concerns or anxiety, or perhaps people just saying that they don't plan to vaccinate, how do you use those data then to make a difference?
Yeah, I think that's another great question. And what our goal is in these populations that we're seeing are significantly more hesitant about getting a vaccine in the future than others is to really have targeted intervention. So presenting the most accurate information. One of the things we did also survey about is how, what the source of information for people in the community is. So are they relying on the Department of Health for their COVID or SARS-CoV-2 information? Are they relying on social media? And so really trying to understand if there's also trends in, if a population is more hesitant about the vaccine, how to give them information at their level or how they process information, so that we can present the most accurate information and reassurances once we know there is a safe vaccine that we could really hopefully get into those communities. And the other way to do this is to really work with community partners. So if we also have like geolocations, we can map this with like the Childhood Opportunity Index. So seeing where there's more vulnerable populations, and get into the communities to really also try and have people from within the community also help emphasize the importance once we have that safe vaccine.
Yeah, that's great. You know, just for everybody to remember, we have our Healthy Communities has some tremendous opportunities to go into the community with mobile health vans and actually help to, whether it's deliver specific messages, to do health maintenance for people and find out what their health state is, or to even do things like vaccines. So Lurie Children's really has some really outstanding opportunities to help deliver care to these communities that may not have as good of access, but we have to know who they are. So that's great. Dr. Coates, what do you know, it's so impressive when you see that sort of those fingerprints, those signatures from, for instance, your RSV study. It's so clear who those kids were who were going to just take their time to recover out of the ICU and probably scare the wits out of everybody taking care of them, versus the kids who are going to make a quick turnaround. I know we're anxiously waiting for your data with the COVID-19 infected children. What's the next step? What do you do with this information when you start to get those signatures? Where do you see that going, and how could that help you take care of patients in the future?
I am so eager to get a look at this data, and I think it's going to be really interesting because we have, you know, data from RSV and flu that we'll be able to compare to the kids with SARS-CoV-2 and see, and then the adults that we've enrolled, to be able to see, you know, can we define a protective signature? Can we define a signature, you know, that classically in RSV leads to a very sick child, and compare that to the signature that we find from SARS-CoV-2 that does not lead to a severe illness. And again, then compare that to adults, and we're hoping to partner with some of our colleagues across the street who are going to start doing the scrapings in the nose on adults who are admitted. Because by doing that, our goal would be to be able to filter down to the pathways that may be most important for and most associated with a good recovery versus progression of disease, because those, similar to what Taylor said, helps you hone in on what pathways might be targeted. A lot of these innate immune pathways have drugs that have been developed for other diseases, autoimmune diseases, et cetera. And so it can help us hone in on what's the protective response that we would want to promote and what's the harmful response that we would want to dampen down.
Great. Yeah, that's really would be an amazing opportunity.
I think the
Dr.
other
Heald Sargent. Oh, go ahead, Dr. Coates. No, please.
Sorry. The other thing is that you could also potentially find, like, the pie in the sky would be, you'd find the 10 genes that matter the most, and then you'd be able to test that, you know, on presentation and say, I'm, you know, this much confident that you're going to be fine in a couple of days, and versus I'm really worried that you're going to progress. And so you could use that information for how you manage the child, whether or not you admit them to the hospital or send them home, that sort of thing.
Yeah, so this is a great example for everyone of what we call personalized medicine. So precision medicine or personalized medicine, where we take that individual fingerprint and we make decisions based on what that person is doing at that time. Dr. Heald-Sargent, it's been really interesting of why children are much less affected by COVID-19 infection when they get even mild symptoms. It doesn't get as severe as often, although it can, compared to adults. What about the adults who don't get sick? Do you think, are they more like just children's responses, or do you think it's just it's going to be a different story?
Yeah, it will be interesting to see. And there have been some reports out comparing those milder adult infections to more severe adult infections. And in fact, looking at their immune system, there seems to be differences in those immune responses. We have to still look at the children to figure out if the mild are the same as the children, and that there's one just phenotype of the immune system that's protective or not. But so far, just how Bria was mentioning, looking at kind of early immune responses in adults, you seem to be able to cohort or group the adults early on within the first few days of infection, looking at their immune system and say they probably are going to get severe. Now, that's not in real time yet, and not all the kinks of exactly which parts are the most important have been figured out, but there have been several different studies in adults that hint that that's going to be possible, that we will be able to tell this is a group that most likely will be milder of infection based on their initial immune response.
And along those lines, do we have the tools, the drugs that if yours and Dr. Coates' research shows, who's going to be more severe? Do we have ways to actually change the immune system early on to sort of maybe put people back on course?
Yeah, in certain instances, it'll depend on where the deviations, where the immune system is going wrong. And as clinicians, we've actually kind of taken some shots in the dark almost and tried some of these immunomodulators that seem to be the most promising, such as targeting, some people might have heard of targeting IL-6 with something called tocilizumab, which is something that clinically we tried to do, and there have been studies into looking at. And as Dr. Coates mentioned, there are other therapies for other inflammatory conditions such as TNF-alpha inhibitors or IL-1 beta inhibitors, other markers of an overexuberant immune response, that if we knew that was the thing that made the difference, there are already approved drugs that we could try to target. But right now, we're kind of shooting in the dark, guessing maybe this is the most important, maybe it's this one. It'd be really nice to know off the bat what's the most important to target.
Yeah, terrific. Dr. Rowley, I think for many people, it's kind of a fascinating idea that you can make antibodies in the lab. So if you all find an antibody that looks like it binds to the virus really well and perhaps would be the right thing that would block the virus, is there a precedent for having sort of synthesized or lab-made antibodies be given to people to give them protection in place of a vaccine, or maybe in people who can't make antibodies well for themselves?
Yes, that's a great question. So for years, we've used an antibody that was made in a lab to help protect certain babies with risk factors from getting severe respiratory syncytial virus, the RSV virus. So that's one example. Some of the most successful treatment for Ebola has turned out to be a cocktail of more than one such antibody made in the laboratory. So there are other examples as well, but yes, antibody therapies are certainly emerging as new tools now that it's a little bit easier to make them in the laboratory than it ever was in the past.
Great, that's terrific. And I'm going to follow that, Dr. Rowley, with there's a question from somebody listening, and I think this will be probably our last question here. But people really have heard a lot of news and discussion about the development of a vaccine and really the discussion's been around adults. What's the pathway for children to get a vaccine? Is that different than adults? And will children likely be able to get a vaccine in the same time that that adults do when one eventually is proven safe and effective?
Yeah, that's a great question. Anybody who read the Tribune this morning may have seen a report about one of our former fellows, Evan Anderson, actually wrote a piece in an infectious diseases journal about how he thought it was bad that there weren't very many ongoing clinical trials in pediatrics for the vaccine, and it was pretty much focused on the adult right now. And I agree with him to a certain extent, but I also do agree with a response that someone interviewed a very prominent vaccine expert in our pediatric infectious diseases field, Dr. Offit, who said, yes, he understood where Dr. Anderson was coming from, but it is true that frequently we test vaccines in adults first before we, unless it's a pediatric only condition or something, before we try it in children. And I think because adults are the ones who are most severely affected by COVID-19, that's another kind of reason why we've been skewed more towards testing in adults to begin with. So yes, I think as soon as there is a candidate or candidates that we have confidence is showing really good results in adults, then we need to do those pediatric trials as soon as possible, because it could be very useful not only to protect the children, but to enable them to not be able to transmit to their grandparents and other high-risk individuals they come in contact with.
Yeah, that's terrific. And I'll just add that so everyone knows, Lurie Children's has actually been working with some of the manufacturers who are developing these vaccines to be one of the sites that will help test some of the vaccines in children as well. So we're excited to be part of those studies as well. Well, I think we have heard some terrific presentations and worked our investigators pretty hard right now with all these questions. So I'm going to turn it back over to Kathy Graham to close out our session here.
Thanks so much, Pat. And, I'd like to thank you for certainly the creation of the Springboard Awards and all the work you've done in mentoring these exceptional, high energy, passionate, dedicated investigators, Doctors Kuzma, Coates, Raleigh, and Heald Sargent. You really are a great inspiration to us. I also want to thank the research champions who you saw earlier, and perhaps you can see again on this page. And I would like to encourage all of you to consider joining us on the second and third calls that are listed here to learn more about what's going on in asthma research as well as adverse childhood experiences. And finally, I want to turn it to our exceptional board chair, Christy Stathis, to say our final goodbye.
Kathy, thank you so much. And thanks to everyone for joining the meeting today. I just wanted to end with just a little bit of background on the Children's Research Fund. I don't have to read the slide to you, but we are a group of 70 board members who truly believe that the private funding of pediatric medical research is essential. And to the extent that you've enjoyed what you've learned today, and you'd like to learn a little bit more about our organization, I would encourage you to reach out to us. We would be very happy to talk with you further. Kathy, thank you. Pat, thank you. All of you from the research team, thank you very much for joining us today. And I hope you all have a great evening. Thanks.